Okay, well, welcome to tonight's webinar. I'm Dr. Jenny Tullett, and I'm going to be talking to you about some of the things that I teach about and research about at the School of Biosciences at the University of Kent. And here in this little picture here, you can see the Biosciences building. Unfortunately, you can't visit today, but hopefully you'll be able to see what it looks like one day. And actually, my lab is here. Just up here in these two windows in the corner. Okay, so I thought it might be interesting before I start talking about some science to tell you how I got to the job that I'm in at the moment. And actually I have a BSc from Kent as well. I did a BSc in biochemistry here. I graduated in 2001. And as part of my biochemistry degree, I also did an industrial placement at Roche Pharmaceuticals, and it happened to be the company branch in Germany that I did that at. And we offer these sandwich placements on all of our BSc programs. And during the time that I was at Kent, I did lots of interesting things. I learned lots of new things. I met really nice, interesting people, and I got the chance to travel as well. And really, it was a combination of all of those things together that made me stay in research and learning for the rest of my life so far. So after I did my BSc, I went on to Imperial College in London and did a postdoctoral degree, a PhD. And then after that, I went to work at Harvard Medical School and did some more research in a lab there. And then afterwards, I came back again to London and worked at University College London and did another postdoc. And then after 9 years of that, I came back to the University of Kent, where I started as a lecturer in biosciences. And there's two parts to my job. Part of it is that I'm a researcher, so I'm answering novel scientific questions. And the other part of my job is that I'm a teacher. So I lecture on a number of different undergraduate courses here. I'm also the director of the BSc in Biology programs of study, which are all accredited by the Royal Society of Biology. And if anyone's interested in what my research is after today's talk, you can see that I have a website and you're welcome to go to that. So my lab works on worms. So I'm going to show you, I've shown three pictures here, A, B, and C, and I should be able to launch a poll now, which is going to allow you to vote for the one that you think that I work on. Okay, is it A, is it B, or is it C? So vote now. Great. Well, thank you all for voting. Well, some of you obviously have a sense of humour, as you've said B, which are the sweetie worms in the middle, which I have some in the kitchen, but that's not what we work on in the lab. A is obviously an earthworm, and it is actually C is the right answer of the ones that we work with. Okay. So we actually work with this third group of worms here called Sonorrhabditis elegans. Okay, and these are tiny little things. This is the lab mascot, which is a bit bigger and stuffed, but these are the worms that we work with. And actually, even this big one here in the middle, it's big because it's taken under the picture is taken under a microscope, but that is actually only 1 millimeter long in real life. Okay, and this is an adult worm. And inside this adult worm, you can actually see just about some embryos. Okay, and each adult worm during its life lays around about 350 of these embryos onto the plate, and you can see those, they're these oval shapes here. And these embryos hatch and they develop into larvae, and the larvae go through 4 stages, L1, L2, L3, and L4, before they come to be an adult themselves. And they're hermaphrodites, so they're self-fertilizing organisms. Okay, they both produce eggs and sperm. And they're made up of 959 cells. And the position and the function of all of these cells is known, which is quite amazing for something that is quite complex, even though it's very small. There's 20,000 protein coding genes in the worm. So that means there's basically 20,000 genes that make something that is useful to the worm. Okay. And of those, about 75% of the worm's genome is actually conserved to humans as well. Okay, so things that we do in the worm and the way that we use this worm to understand scientific questions is actually highly relevant to human biology as well. And this is a nice, very schematic textbook image of a hermaphrodite worm. And here you can see the eggs drawn in the middle just before they hatch. Okay, and this is the head of the worm here, and this is its tail. Okay. But this little worm gets an awful lot of attention. And actually, in the last few years, it's got 3 Nobel Prizes to its name. Okay, the first one was in 2002, the Nobel Prize for Physiology and Medicine. And that was given to Sidney Brenner, who was the man who actually decided to start using this worm in the first place for scientific research. And he got this prize along with John Sulston and Robert Horvitz for their discoveries on looking at how different tissues or different organs in the worm were actually developed. Okay, and then in 2006 there was another Physiology and Medicine Nobel Prize where Andrew Fire and Craig Mello got a prize for their discovery of RNA interference. And this is a fascinating genetic trick for playing around with the genome of both C. elegans and all sorts of other species as well. And if you're interested in this, this is something that you would learn about during your undergraduate degree at the University of Kent. And then again, in 2006, there was a chemistry Nobel Prize for Martin Chaufey and his colleagues, and they got their prize for looking at the green fluorescent protein in the worms and discovering green fluorescent protein. And that's something that was originally identified in jellyfish, but it is now used as a biological tool in a wide range of systems. And I'm actually going to show you some really nice scientific images that use this later on in my talk. So in the lab, these worms are really, really easy to use. We grow them on these little plastic plates, Petri dishes, which have got a bed of agar in them, okay? And then on top of that agar, there's a splodge of bacteria, okay? And the worms basically just crawl around in that bacteria. That's why you can see all these kind of tracks and squiggles on this plate here, because that's just the marks that the worms make in their food as they crawl around on it. Okay, and they eat the bacteria and they live on it quite happily. You can move worms from plate to plate with something called a worm pick, which is basically just a stick with a little bit of wire on the end, and you can pick each worm up individually. And because they're so tiny, we need to have a look at them underneath the microscope to be able to do this picking and actually be able to tell anything about them. Okay. And this is a little video of what they look like down the microscope. This is speeded up a little bit, but people often say when they first start working with them in the lab that this is what they see when they close their eyes and go to bed at night, is they can just see hundreds of little tiny animals crawling around in front of their eyes. So you can use this organism for all sorts of things, as you can see by those examples of the Nobel Prizes that I gave. But at the moment, one of the things we're doing in my lab is to think about the question about why we eat or when we eat, and how do we know when to stop eating? Okay, so I'm going to try another little poll now and ask you another question. Okay, so here we go. So the question is, how do animals decide when to eat and when to stop eating? Okay, you've got choices. You've got, is it smell, taste of food, sight of food, signals from the brain, signals from the digestive system, or all of these? So see what you think. Any more answers? No? Okay, I'll end that now. Okay, can share the results with you. So the answer is actually, as most of you got right, was actually all of these. Okay, and we'll talk about this in a moment. Okay, so we're using these worms to ask the question, how do animals perceive or sense or react to food? And this is really important in biology because these kind of responses have implications for a whole range of different metabolic disorders. So things like obesity or eating disorders or diabetes, all of these things can be impacted on how we react to food. So as you were quite rightly saying in the poll, there's actually a whole range of different things that are required for a body to actually be able to sense food. Okay, so you obviously smell food, you taste it, you see what it looks like, and that tempts you to eat it. But it's far more complicated than that, really, because you actually also have signals from your digestive system, which are known as gut peptides. So things like PYY and GLP-1, all sorts of things like that. Ghrelin you might have heard of as well, and they come up from the gut. You've also got signals from other tissues. So for example, your fat tissue, you might recognize the hormones insulin or leptin, you might have seen in stories in the news or learned about in school. But all of these signals are actually integrated in a part of the brain called the hypothalamus. Okay, and this is required for regulating a process called satiety, which is basically how you know when to stop eating. Okay, and all of this stuff comes together and is processed by the brain. And we call this neuroendocrine control. Okay, and this sounds like a bit of a fancy word, but you can basically break it down as you can with most biological terminology. So neuro basically means brain and endocrine means hormone. Okay, so you've basically got brain hormone control of learning how to eat and when to stop eating. And it's actually possible to study this in a worm. And one of the things we do is we study the genetics of it. So the genes that we have in our bodies that are responsible for controlling food-related behavior. And we can do it in something simple like the worm because the worm does actually have a few different simple, measurable, scientists like things that can be measurable, food-related behaviors. It's got a genetic system. We know all the genes in its genome, the 20,000 protein coding genes I was telling you about, and also its whole nervous system. So all the neurons in its body have been mapped. We know where they are and we know what most of them do as well. Okay. And the work I'm going to tell you about today was the work of Nick, who's a PhD student in my lab, and Max and Alex, who are also students in my lab a little while ago. And actually, it's interesting to see that both Nick and Alex were actually undergraduates at the University of Kent before they started their work with me. Okay, so if we're going to use the worm to study food-related behavior, it's really, really simple. You've basically got food intake. So at the mouth of the worm, the food goes in. There's a whole range of chemosensory neurons. These are kind of neurons that sense food or sense the food in the environment before it comes into the worm. There's endocrine signaling. So things like insulin hormone, they signal to the rest of the body of the worm. And all of this comes together again in the nervous system to control the behavioral change of the worm. And those behavioral changes in turn feed back to control the food intake. And worm genetics is really very simple. We learn about it in our degrees, but there's really only two concepts you need to grasp to understand it. The worm is a really good genetic model. Okay, and this is a way we use terminology of just saying that something's really good with doing genetics. You can do really good genetics with it. Okay, so you can do two things. You can either knock genes out, so you can take genes away, or you can add genes in. Okay, and by doing that, you actually learn an awful lot about what different genes do. Okay, and then you can get a little bit more fancy if you want to, and you can actually knock different genes out at the same time as each other, and you can actually find out how genes work together as well. Okay, but for the sake of today, you need to know is that you can knock genes out and you can stick them back in, and that tells you what the gene does. Okay. So next bit of knowledge that you need to know is that worms have different food-related behaviors. Okay, they basically do three things, right? They roam around a plate, they look for, and while they're doing that, they're looking for and eating some of the food. Or they kind of start to sit still and they do this thing called dwelling, where they move back and forward a little bit. Okay, and that's when they're eating the most of the food. Or they do something called quiescence, which is basically sleeping. And this is the thing they do when they're completely full. Okay, so it's related to human satiety. Okay, and we can measure these things in a worm really easily. Okay, we've got a lovely assay that we do. You stick one worm on a plate of food, okay, before you go home in the evening. Okay, and then you come back 16 hours later, right? And you take a photograph of the plate and you can see all the places that the worm has visited on that plate overnight. Okay, so this is the nightlife of the worm. And then you can put that photograph onto the computer and you can put a grid over the back and you can work out what percentage of the plate the worm has walked over. Okay, it's really neat. And this is the kind of picture that you get when you take these images. Okay, so this is a normal worm. Okay, this is what a normal worm does. And a normal worm covers around 70 to 80% of the plate overnight. Okay, so we've been interested for a very long time in our lab in a gene called SKIN1, SKN1. Okay, and this gene is a transcription factor. Okay, so what that means is that SKIN1, this gene, once it's been made into a protein, it can stick to the DNA. Okay, and what it does is it switches on other genes. Okay, and together, all these genes that it switches on do all sorts of interesting things in the body. Okay, they're involved in the stress response. They can balance the redox state, so the amount of oxygen that's available. They control fat, mitochondria function, immune responses, and a whole range of other different things. Okay, and as a result of that, SKIN1, this gene that we're interested in, is really important for regulating lifespan. It regulates stress resistance. And for the purpose of today, it's really important for food sensing behavior. Okay. And importantly, this gene is also controlled a little bit by the environment and specifically the diet that the animal has. And it's interesting, not just because it's a gene that we're studying in the worm, but because mammals also have a version of this gene, and it's called NRF. And obviously in an ideal world, the gene name would be exactly the same in the worm and a mammal, but unfortunately that very rarely happens in science, okay? So you just have to remember SKIN1 is the same as NRF in mammals, okay? So we're interested in this gene and its role in food-related behavior. Okay, so there's 4 things called isoforms of this gene. So that basically means there's 4 bits of DNA in the worm that code for this gene. And they're all slightly different sizes. That's all you need to know. And we're interested in one of these, this little one here, okay? And remember our genetic model, okay? So you can knock genes out and you can put them in, okay? So if you knock genes out, we basically make a mutant, okay? And we have a SKIN1B mutant. And that mutant basically means that you've taken out a chunk of DNA, okay, in this isoform here. And that means that this gene can't be made anymore, okay? So that's a mutant version. And that allows you to understand what the gene is doing. And you can also add genes back in. And if you add them back in, you can do something pretty cool. You can basically just tag them with green fluorescent protein from those jellyfish I mentioned earlier, okay? And if you tag this little bit of gene here, this SKIN1B gene with green fluorescent protein, you can see that you get these 2 little spots in the head of the worm, okay? And these are really cool. And they're called the ASI neurons. It basically looks like 2 eyes, actually, when you're looking down the microscope. But because we know the structure of the whole nervous system of the worm, we know that these 2 neurons are actually called ASI neurons, okay? And these neurons are known to be chemosensory. So that means they are actually capable of detecting food in the environment. So that was a really cool experiment to do this, because we actually know where this gene is in the worm. And often when you know what bit of the worm something's expressed in, it tells you a little bit about what that gene might do. So for us, that was giving us a clue that it might have something to do with food type of behaviors. Okay, so the question we asked was whether this gene was actually controlling behavior in the worm. So it's only in these 2 neurons, right? And if you've got a normal worm, this is what happens if you put a worm on a plate overnight. Okay, it's quite busy. You can see it's been roaming around quite a lot. However, if you have a type of worm that doesn't have that SCIM1B gene in anymore, look what happens to its behavior. Look, it can only cover about 40% of the plate, and all its little tracks are really convoluted and tied up together. Okay, so why do you think this is? Okay, let's try the poll again, see if I can do it right this time. Okay, so third poll. Okay, so why do our mutants move less? Is it because there's something wrong with their muscles? Or is it because they show different behavioral patterns? Or do you think it might be both of those things? Okay. All right, well, it says 50-50 split between people that think that it's because of the behavioral patterns and people that think that it's both of these things. So the answer is that it could actually be both. And again, let's see if there we go. So it was really important because it could have been both of those things. It might be that the worms couldn't move very well, or it might have been because they had different behaviors. Okay, so it's really easy to test whether worms move well. And to do that, you basically take them for a swim. Okay, so you put the worms in a drop of liquid. Okay, and you measure their ability to move. So we call it thrashing. So the ability to move back and forward, okay, in a period of time. And we did this with some normal worms, and we did it with some SCIM1B mutant worms. And this basically tests the worm's muscle activity. It just tells you whether it's capable of moving as well as a normal worm or not. And what we found was that actually there was absolutely no difference between the activity in swimming motion of wild-type worms with SCIM1B mutants. So those SCIM1B mutants are not exploring the plates, not because they can't, but because they are choosing, for some reason, not to explore that plate. Okay, so there's something going on with their behavior. Okay, so it must be affecting these behavioral patterns. So that means it's either affecting roaming, dwelling, or sleeping. Okay, and again, all of these things are linked to food intake, the chemosensory neurons, endocrine signaling, and eventually to the behavioral changes. Okay, so one of these behaviors that it's really easy to measure in the worms is the sleeping one, the quiescence one. Okay, so what you do is you basically take all the food away from the worm overnight, so you starve it. Okay, and then in the morning you refeed it again, and you give it a really nice rich diet. Okay, so it's really happy, it has a massive breakfast, and then you give it 3 hours to recover, and then you find that it will go to sleep. Okay, for a longer period of time, and you count the number of seconds that it's sleeping. Okay, and if you do that, you find that you can count periods of time in a normal worm, so the black bar here, that are around about 15 seconds at a time. But in the SCIM1B mutants, it's pretty much double that. Okay, so these are very, very sleepy worms, and these are simple assays, but they do take quite a long time because you have to sit and look down the microscope for quite some time, counting seconds that worms are asleep for. But this told us something really interesting, and that was that there was a difference in the type of behavior that we get in between a wild type worm and a mutant worm. Okay, so we've basically got a strain of worm that is very sleepy. And importantly, this tells us something about what that gene is actually doing. Okay, so if you take away that SCIM1B gene, okay, you've got more sleep or increased satiety. So if you're then thinking about the function of that gene, that's basically telling you that that gene is needed to stop sleeping or satiety when it's in the worm like it normally should be. Okay, so that was really interesting. But then during these studies, we also noticed one more really interesting thing. And I just want to tell you about that before I finish today. And that was that if you look at a normal plate of worms, so a normal population of worms, you'll see that some of them are on the food and some of them are wandering off the food. And that's because worms like to roam off and look for something better. Okay, and then they come back to the food, but they go off the food and come back to it. But our mutant strain's a bit odd. All the worms are on the food all of the time. Okay, so they don't roam off and they don't roam and look for different things. And we wondered whether this was something to do with the fact that the worms kind of thought that they were hungry all of the time. Okay, they couldn't really sense the outside environment properly, so they couldn't tell whether they had enough food or not. And so they just decided to stick around on the lawn the whole time and just keep eating. Okay, so they might have a problem with that. So there's a bit of the cell that you can look at when you're thinking about energy levels and sort of, regulating the amount of energy that a cell or an organism has to spend and why it might be wanting to eat more or not. And that's the mitochondria. Okay, so the mitochondria are these little tiny organelles here. It's part of the cell. Okay, and you'll learn all about all these different parts of the cell when you come to the University of Kent to do an undergraduate degree. So you'll have done some of it in school, but we'll do a lot more of this at university. But today I'm just going to talk to you about this one little organelle called the mitochondria. Okay, final poll for the day. So mitochondria, what do they do? Are they important for storing genetic material? Are they important for making energy? Or do they make protein? Okay, great. Well, most of you got the answer I was looking for, which was to make energy. One person actually said that they store genetic material, which is completely correct. There is some genetic material in the mitochondria, although that wasn't quite what I was getting at for today's purposes, but you're absolutely right. Okay. So these mitochondria are really energy powerhouses. What they do is they generate energy that the rest of the cell and the body can use when they need it. Okay, and as a result, they can help animals deal with changes in the amount of food in the environment. Okay, and to do that, they do something that's really quite clever. They can join themselves together. Okay, and if they join themselves together, they can actually increase the amount of energy that they put out. Or they can split themselves up and they can fission themselves apart and make themselves small again. And that can actually also, that can reduce the amount of energy and also help with quality control of the mitochondria in cells. Okay. And so we decided to have a little look at the mitochondria in our SCIM1B mutants. And what we found was this is electron microscopy. So we're looking really, really, really close up at our worms now. And you can see some muscle tissues down the side. And then these blobs here are mitochondria. And this is a normal worm here. So you can see some nice round-ish mitochondria like you would expect to see. Okay. But if you fast a worm, so if you take its food away, you can actually see that the mitochondria start to join up here like this. Okay, so they're trying to increase the energy in the muscle. Okay, so they can help the worm survive these low food conditions. But the weird thing was that in our mutants, in our SCIM1B mutants, the mitochondria are massive. They've already fused together to make this massive network. So they probably think that they're a bit starved. Okay, so something is not right with this mitochondrial network here. And we wondered whether this might have anything to do with the different behaviors that we were seeing. So whether the worms decide to roam or dwell or go to sleep and quiesce. So we did a clever little genetic trick. And if you look at the SCIM1B mutants under normal conditions with their fused, big fat fused mitochondria, you can see that the animals are not moving very much. But when we use this, we can use a little genetic trick, okay? And we can actually break the mitochondria up, okay? And when we break the mitochondria up, we completely rescue or start the worms being able to move all over the plate again. So this weird not moving, extra sleeping behavior is all due to the fact that the mitochondria are joined up together like this. Okay, so this was really interesting, and we're now looking at this a little bit more in our lab. So let's wrap this up. What have we learned this evening? You've learned a little bit about how to use worms as a scientific tool. You've learned the basics of neuroendocrine signaling. You've found a new gene that's involved in controlling satiety or sleeping behavior in a worm. And you've learned that mitochondria can also be linked to controlling behavior. And all of this works in a kind of pathway, okay? And you've got the environment linked to the nervous system, linked to other tissues in the worm, linked to a physiological response, which is the behaviors we've been talking about all along, okay? And although we've done all of this in a worm, there's a really exciting possibility here that those mammalian genes that I was talking about, the NRFs, might actually be involved in regulating food-related behavior in us, in our brains. And actually NRFs are expressed in the brain. So that is actually a very real possibility. And that will be something that we start to look at more in the future. So this could be you working in a research lab at Kent doing this kind of thing. We've published this, well, we're in the process of publishing this work now. This is the study that we've done. And I thought it would be interesting to show you that actually during your time at Kent, in your final year here, you do an 8-week research project in the lab where you're teamed up with a member of academic staff. And I run these every year. And back in 2017, I had 8 students in the lab, but 2 of them, Alexander and Timo, were working on Skin 1 with me and this project. And actually both of them produced work during that 8-week period that actually fed into this publication that we're doing now. So obviously it takes a few years for these things to actually mature and come to fruition, but it was fantastic that these undergraduate students really could contribute to some real scientific work that we were doing in the lab. And I really love that part of my job, that I can bring together the research and the teaching so well. So the last thing that remains for me to do is just to thank everyone. So this is my group. Nick has done most of the work that I showed today, but there's also some help from Max and Alex. We collaborate a lot with Dr. Marina Escura, who also has a C. elegans lab at Kent, and she runs the neuroscience course in 3rd year, if anyone's interested in that. And we also collaborate with people at University College London and at Harvard's Joslin Diabetes Centre, as well as the other university in Canterbury, Christchurch and King's College in London. So thank you very much for listening. And if you have any questions, I'm happy to take them. Thank you. Is it just us?
I think so. Oh no, we've got a question. Can you see the chat?
No, hang on, I can close my, no, I can't see it. Just read it out for me, would you, Alana?
Yeah, that's right. So from Alex, how did you know when the worms were sleeping?
They basically don't move. That's a really nice question, actually. I should have explained that better. So they basically don't move. They just sit very, very still on the plate. You know they're still alive because you can see their mouth moving a little bit, but they're basically just still. So you just have to look very carefully down the microscope. And actually my student gets quite protective of the lab space when he's doing these experiments, because the worms are very sensitive to any movement that goes on around them. So he likes everything to be very quiet and still while he's doing these experiments so that the worms can't be disturbed. Does that answer your question?
Yes, it did. From Oliver, thank you for the excellent presentation, Dr. Tullett. Are there any ethical considerations with C. elegans?
Thank you, Oliver. It's very kind of you. Very good question as well. I guess there's always a little bit of ethical consideration to be had whenever you're handling an animal in the lab. but there are no home office requirements, for example, for using scalligons. So the numbers we use, we treat them very much like we use bacterial culture or a yeast culture in the lab. So no, there's no major ethical consideration that has to be taken into account for using either worms or Drosophila, actually, so fruit flies as well. Nothing like you'd have to do with a mouse.
They said perfect, thanks. Haven't got any more coming through at the moment. But I will just say that was one of my favourite practicals that I did, was the worm picking.
Oh, did you like that? Good.
Yeah.
We're running it this year as well, actually. Even with the lockdown practicals, we're actually doing a mini picking, a picking practical, basically, as well as we can still do the one that you did. So the RNAi practical, we do that still, but that's going to be online. But we're actually going to have some lab work as well with the worms, because I think it's quite good fun.
Yeah, it takes a while to get the technique, but then the moment you get the technique, you can work so quickly. It's great. I loved it.
Yeah, well, I still like it too.
Did anyone have any general questions about university or studying at a university?
Yeah, James and I are both current students. You are a current student, aren't you, James?
Yes.
Just double checking. And that's if you've got any questions about student life or like why we picked the uni or anything like that, then you're more than welcome to ask those as well.
So I'm currently a research master's student with Jenny. So I'm kind of doing the projects you just talked about, but a bit more neuroscience-y. I want to find out, what the neurotransmitters are firing and things like that to allow SCIM1B to do its function. So I don't really know the background of anyone here. So if you hear like the word masters or undergraduate and you're a bit confused, so a research master's is basically you work in the lab like a normal job. Whereas if you hear a taught master's or like an undergraduate degree, you might go to lectures or seminars where you get taught information, a bit like at school where a teacher will teach you stuff. A research master's, you just get full practical experience. So you're always in the lab. So if you think, oh, actually, that sounds interesting, I'm more of a doer rather than a sit and listen to someone, maybe look into that for the future.
Yeah, I think they're great, actually, research masters. I think they teach you a lot. You learn all the backgrounds as well as how to do stuff.
Yeah, and they're much cheaper too.
And I am currently a fourth year undergrad student. So I spent the first 2 years studying for my undergrad, and then in my third year, I went out on placement to Canterbury Hospital and worked as an assistant healthcare scientist for a year. And I'm now currently in my final year, just starting my project as well, which is quite fun. I'm doing one of the new ones this year. It's the outreach, and that's so just completely brand new, and that which is fun.
Great.
And, yeah, it's something else I really liked as well. The fact that a lot of other unis I went to, it was either just dissertation or lab. Whereas here, there's like 6 different types of projects you can do for final year, because you've got like bioinformatics, business, communication, outreach, on top of the normal ones as well. So a lot more choice.
Yeah, lots of choice.
Are you biomedical science, Alana?
Yes, I am. Yeah. And I hope to become one, actually. I did my portfolio while I was on placement.
Oh, well done.
Yeah, thank you. And that's so when I graduate, I will be able to register, and I'm going into the working world. I'm ready to go and leave education now. But yeah.
Well, thank you all very much for coming this evening. I'll stop recording now. But if you do have any questions, please feel free to get in touch with either myself or anyone else at the recruitment team at Kent. Would always be very happy to answer your questions. So thank you very much. And
I'm also on UniBuddy, so if anyone wants to send me a message about student life or anything like that, then if you search up UniBuddy on the University of Kent page, you can contact me that way as well, just to let people know.
Great. Thank you, Alana and James.